Hello, good morning students. Today we'll discuss about the Obelia colony. It is an alien trait. The colony of Obelia consists of horizontal thread-like root called hydrorhiza, which is attached to the weed or any substratum. From the hydrorhiza arise vertical branching stems known as hydrocoelis. Hydrorhiza and hydrocoelis are the hollow tubes. The hydrocoelis bears zooids or polyp on either side in a cymose formation. At the growing end of the main branch are immature club-shaped polyps. Each polyp has stem and is having a terminal head called hydranth. The hydranths are the feeding polyps. They feed by capturing minute animals and larvae. Towards the base of the hydrocoelis is in their axil of the polyp reproductive polyps called the blastostyles. They are present. The polyp, their tubular connections and blastostyles are made up of ectoderm, mesoglea and endoderm. These layers are together called coenosarc, and its cavity in the enteron, which is continuous with the and common with all the members. Through enteron, digested food is distributed in solution. The entire colony is covered by tough yellow chitin secreted by the ectoderm, and this covering is known as perisarc. The perisarc constitutes the exoskeleton, and it covers the hydranth, hydrocoeli and their branches, and at the base of each polyp it forms clear wine glass-shaped hydrotheca. The hydrotheca has a shelf across the base which supports the hydranth, and the hydranth can contract and withdraw into the hydrotheca. The perisarc around the blastostyle is a gonotheca. The blastostyle and gonotheca are together known as gonangium. The perisarc is an exoskeleton. At first it is continuous with the coenosarc, but on going thick it separates and joins to the coenosarc only at intervals by minute projection. At these places it gets ringed, forming annuli, which allows the bending of the colony. The Obelia is trimorphic colony and is having three kinds of zooids, namely polyp or hydranth, which are the nutritive zooids, the gonangia or blastostyle. They are the budding zooids, MEDUSA, the sexual zooid. The colony of Obelia has many polyps and hydranth or the gastrozooids. It is a cylindrical body attached to the axis of the hydrocoelis by its proximal end and free at its distal end. It is covered by cup-shaped hydrotheca. The free distal end is produced into a conical elevation, the hypostome or manubrium, which is about 1/3 of the length of the hydranth or polyp. The hypostome is surrounded by circle of numerous tentacles, about 24 such tentacles they are present. The tentacles are longer than the hypostome and tapering and are filiform. The apex of the blastostome appears to be having the terminal aperture called the mouth, which is capable of great dilation and contraction. Below the hypostome is the stomach region of the polyp. The body and the manubrium of the polyp enclose a spacious enteric cavity called gastrovascular cavity. The polyp is protected in hydrotheca, which is prolongation of the perisarc. At the base of the polyp, it forms a ring-like horizontal shelf on which the polyp it rests. The tentacles, they are solid, having large number of nematocysts. The polyps have an outer layer, ectoderm, and the inner layer, the endoderm. Between these two, a transparent mesoglea is present. The enteron has fluid and its lining is flattened, flagellated. The rhythmic contraction of the hydranth cause a current which distribute food obtained by some polyps to those parts of the colony where feeding is not taking place. The tentacles are solid and have no enteron. Ectoderm, it consists of long, conical, columnar epitheliomuscular cells. Their inner cells are produced into muscular processes which run longitudinally. In the ectoderm layer, few interstitial cells, some branching nerve cells, and cnidoblast, they are present. The nematocysts are abundant on the tentacles and manubrium only. The nematoblasts are found at the basal part of the hydranth and in the cenosurk. The Obelia has only one kind of nematocyst called the Basitrichus isorhizas, in which the capsule is oval but is absent and thread is open at the tip and has spines on its base. The endoderm, it has long granular epitheliomuscular cells with muscle processes point outwards and are circular. Endodermal cells have flagella which produce a current in the enteron. They can also form pseudopodia for engulfing the food. The endoderm of the tentacles has cubical vacuolated cells with thick walls. In the endoderm layer, nerve cells, club-shaped gland cells which produce digestive enzymes are present. Mesoglea is a thin jelly-like substance with no structure or cells. On each side of the mesoglea is a nerve net composed of nerve cells and their fibers. The two nerve nets are interconnected. The polyps, they are the nutritive food zooids of the colony. It is carnivorous, feeds on the aquatic crustaceans, nematodes, and other worms. Tentacles help in catching and conveying the prey to the mouth. The digestive juices are secreted by the gland cells of the gastrodermal cavity of the polyp, and this process of digestion is extracellular as well as intracellular. So in this diagram we can see the section of the polyp and the nematocyst, the closed nematocyst and the opening of the thread when it is stimulated. Second is the gonangium. The gonangium is club-shaped cylindrical form. It is covered by transparent gonotheca and it contains an axil or blastostyle on which the lateral buds form that develop into medusa or gonophores. The blastostyle has no mouth and no tentacle but ends distally into the flattened disc. The gonotheca opens at its distal end by a gonopore through which the medusa escape. The gonotheca, blastostyle and gonophore together forms the gonangium. When the temperature of the water exceeds 20 degrees centigrade, the buds which would normally form gonangia in the colony breaks free from the colony and settle down. A stolon arises from the lower end of the bud which produces a new colony of Obelia asexually. This is a special mode of asexual reproduction in the case of the Obelia. Now this is the structure of gonangium with gonopore, gonotheca, blastostyle, developing medusa, and the inner is coenosarc covered by transparent perisarc. The blastostyle produces medusa by budding in large number. The cavity of blastostyle pushes the coenosarc out to form a small protuberance or a bud. This bud grows larger and its coenosarc becomes like a vesicle which is attached to the blastostyle by a narrow stalk. The cavity of the vesicle is continuous with the anteron of the blastostyle. The distal ectoderm of the vesicle separates into two layers. Then the inner layer of ectoderm splits to acquire a cavity called the bell rudiments. There are now two layers of ectoderm outside the bell rudiment and one layer on the inner side. The cavity of the bell rudiment assumes the shape of subumbrella and the manubrium is formed in the center. The two layers of ectoderm which enclose the bell rudiment form the outside, now breaks, leaving the marginal and circular shelf called the velum. The manubrium acquires a mouth and the marginal tentacles are formed. The stalk breaks and its aperture closes up and a medusa is formed which is set free. So in this way, in these 6 steps, the medusa is developed on the blastostyle. The medusa is modified zooid produced as a hollow bud from the coenosarc of the blastostyle in spring and summer. The medusa swims freely on the surface water. It is soccer shaped. It is attached to the middle of the convex surface to the blastostyle. When fully formed, it breaks free and emerges from the mouth of the gonotheca. The medusa is circular and tiny umbrella-like structure in shape. The convex outer surface is known as exumbrella and the concave inner surface is known as subumbrella. From the center of the subumbrella arise short projecting manubrium at its arm in the aquatic mouth surrounded by 4 oral lobes. The mouth leads into the enteric cavity or gastric cavity in the manubrium. From the enteric cavity arises 4 radial canals which are delicate ciliated tubes. They run to the margin of the bell to join a ciliated circular canal running near the margin. The enteric cavity and the canal represent the anteron which distributes the food. So in this diagram we can say the mouth is there, manubrium is there and the stomach is present. 4 radial canals they are there and on the radial canal we see the presence of gonads and on the margins the tentacles they are present in the bell-shaped structure. Projecting from the middle of the radial canal are 4 gonads. Since sexes are separate, there are either 4 testes or 4 ovaries. They are patches of modified subumbellar ectoderm. The gonads mature after the medusa escape from the gonotheca. The edge of bell is produced towards a thin fold called velum. From the edge of the bell, numerous small tentacles, solid tentacles, they arise hanging downwards. These tentacles have swollen bases due to the accumulation of interstitial cells, which are particularly absent in other regions. The basal swelling of the tentacle are called the vesicles or the bulbs. Neurocysts are formed continuously in the bells from where they migrate to the tentacles. The digestive enzymes are secreted from the endoderm of the bulbs. There are 8 marginal sense organs called statocysts or the lithocytes, lying at regular intervals, being attached on the subumbellar side of the bulbs of 8 tentacles. They are developed in response to locomotory habit. A statocyst is a tiny circular closed vesicle lined with ectoderm and filled with a fluid containing calcareous granules called otoliths, which lie in a special cell called lithocyte. The lining has some sensory cells with thin sensory process on which the otoliths produce a stimulus which is transmitted by nerves to the muscles. The muscles coordinate swimming movement of the medusa, and when the medusa becomes tilted, the muscles contract to the right, and the position of medusa, bell and statocyst are balancing organs.